Hey everybody, welcome back. We've done our unit opening where we spoke about the marathon runner. We're going to continue speaking about why this young lady's runner 0358 fell into a coma. We have some investigations to do, one of which we've actually already done prior to this lesson, which was the yeast investigation. And the yeast investigation was our first two of our 5 E's for this 5E sequence. So for every sequence or every subunit that we have within the unit, we're going to have an engage where we open up the unit and just introduce some different scientific jargon to us. We have our explore, which is usually some type of lab or investigation that we use to explore a certain phenomena. Then we have our explain, which is what we're going to go over today. And our explain essentially just explains what we did during our investigation, during our explore phase. Our next one will be an elaborate, where we just elaborate on certain concepts that need to be emphasized. And then finally, our evaluate is going to be a performance task. That's something that you're going to do on your own. Don't worry, we'll walk you through it. All right, so our aim says, how can we represent the process of cellular respiration in a cell? Now, keep in mind, we want to remember a lot of the stuff that we did prior to this lesson during the yeast investigation that we've already done. All right, so let's read our do now question. It says, apple cider vinegar, excuse me, sorry. Apple cider vinegar containing yeast was placed in a sterile container and kept at room temperature. Sterile just means that it does not have any microorganisms whatsoever. When small bubbles of gas formed in the cider 2 days later, a student asked, what is producing the gas bubbles? Which statement is a hypothesis that could be tested experimentally to answer this question? Now, normally our hypothesis is in an if-then or maybe if-then-because format, but it doesn't always have to be that way. Your hypothesis just has to be a testable statement. So out of these 4 answers, I'm going to look for which one is a testable statement. I'm going to get rid of anything that is not a testable statement. So if I look at letter A, it says set up an apparatus to collect and test the gas. That's more of an experimental setup as opposed to a hypothesis, right? So that's something that I'm going to do after my hypothesis is made. So I can actually cross out letter A. Now, B says the rate of gas produced must be determined. That's maybe true, but that's not a testable statement. So I'm going to cross out B. Now we're left with C and D. Let's take a look at C. A container of cider without yeast should serve as a comparison. Sure, that's a part of the experimental setup, and while that might be true, it's also not a testable statement. But if we look at letter D, it says cellular respiration is being carried out by the yeast. We can actually test that, right? Which we actually already did test when we measure the height of the foam. So D would be the strongest answer choice here. Now, if we take a look at number 2, it says base your answer to the following question on the information and diagram below and on your knowledge of biology. A student conducted an investigation on fermentation. Yeast, molasses, and water were combined in a fermentation tube and left for several hours at room temperature. The results are shown below, right? So yeast, we should already know, is a unicellular eukaryotic organism that we use to test the rate of respiration. Molasses is just a very, very heavy syrup. And every time that we're talking about some type of syrup, what's the primary nutrient that's found in syrups? Sugar, right? And we know that sugar is definitely used in respiration because it gives our cells a lot of energy that it needs. And then, of course, water, because we know that we need to mix warm water to actually activate the yeast. Okay, and they were left inside of this tube, and the results are shown below. So inside of the tube, we have molasses, yeast, and water, right, at the start, which is in the first setup. And then over a couple of hours, we see that there's a little bit of X towards the top that wasn't present a couple of hours prior, all right? Which substance, sorry about that, which substance would most likely be found in the greatest amount in area X, right? So let's see if we can cross out anything that doesn't make any sense to us, right? Area X, because this is a sealed container, it's likely that there's not a solid or liquid that's being produced here by the yeast. Because remember, this is essentially the same setup that we saw in our previous investigation with the yeast, right? And we knew that they were producing some sort of gas. So I can actually cross out anything that is not a gas. So if I look at A, it says O2. O2 is just oxygen. It's just a symbol for oxygen. Is oxygen a gas? Yes. So I'm actually going to hang on to letter A because it is a potential answer choice for us. B is C6H12O6. This is actually the formula for glucose, which is that simple sugar that our cells absolutely love, and that's used as a reactant during cellular respiration. So I can actually cross that out because sugar is not a gas. Obviously, it's a nutrient. It's a solid that our cells use. C is H2O, which by now we should know is water. And in this case, the water that we have inside of the fermentation tube is in liquid form and not gaseous form. So I can cross that out. And then D is CO2, which is carbon dioxide, which is always in the form of a gas, right? But now I have to see, whatever X is has to be a waste product because it wasn't used initially as a reactant. So it has to be something that was produced over several hours later. We saw from our investigation yesterday that the yeast needed oxygen inside of there because oxygen is used as a reactant in cellular respiration. So the only logical answer that we have here is carbon dioxide because carbon dioxide is a waste product of respiration, right? We exhale it because our body doesn't need it. Same thing with the yeast, right? They get rid of it because their tiny little cells don't need it either. So it's a waste product of respiration. All right, part A, cellular respiration model. All right, so it says use the space below to develop an input-output model that represents what you learned about cellular respiration from data you collected during the yeast respiration lab. All right, so our inputs are anything that we added to the cell or into the bottle during that investigation. So let's just break that down. 1A, our inputs. Let's get our inputs in. All right. So one thing that we know that we added in was the actual yeast itself, right? Which is serving as the model organism. Okay. Another thing that we know that we added were those packets of sugar, those little Domino's packets of sugar. So we have sugar here, which is our glucose. Okay. And remember that our bottles were not sealed. They were actually open to the open air. So we know that oxygen has to be entering those bottles and that yeast are continuously able to produce that foam because they have a sufficient supply of oxygen. So even though we didn't pump oxygen into the bottle, we know that oxygen is constantly entering the bottle. Okay, so we have oxygen, which is O2. Okay, and then we also know that we have water, which is H2O, right? We added in that warm water. All right, so those were our inputs into our bottle, so to speak, right? Now the cell that we were talking about is yeast once again, right? And now it's asking about structures and processes. So what was the phenomena? What was the process that we were exploring during the investigation? Well, if you guessed cellular respiration, you would be correct. All right. So cellular respiration. Okay. Now, what structure, what organelle does respiration always take place in as long as there's a sufficient supply of oxygen? If you guessed the mitochondria, you are absolutely right. I'm going to just draw what the mitochondria looks like. I'm also going to label it. And I'm just going to make like a small caption as powerhouse of the cell, right? This is where cellular respiration takes place. And we know that cellular respiration, right, produces energy in the form of ATP. right? That's why our cells just absolutely love it. Now, as a result of combining these inputs into the bottle, what were the yeast now producing as a result of that, right? So one of the things that we know that they definitely produced was carbon dioxide. We were able to measure that foam and that foam height. Those foam bubbles were carbon dioxide bubbles that were being produced. So we have carbon dioxide, okay? So CO2, all right? And then during respiration, we always release small amounts of water. So there's always water that's kind of going in and water that's also coming out. So water is also produced. But the whole purpose of respiration, right, is to produce ATP energy, which we literally just stated here. That's the whole purpose, right? This is cellular energy to be used by cells. Used by cells to perform work, right? So maybe muscle movement, maybe transportation, right? Different metabolic processes are going to use ATP energy, all right? So those were the three primary outputs as a result of these inputs that were here, okay? Now I'm just gonna make a mark for us that shows, and I wrote this purposely like this, that CO2 and water are waste products, right? Yeast, just like ourselves, we're going to release that CO2. We have no real great use for it. And we can afford to sacrifice some water because we want to make sure that we're maintaining water balance at all times, which we're actually going to speak about later in the unit as well, right? But what I'm holding on to, what my cells are holding on to, is ATP energy, which is not a waste product. This is the purpose of respiration, right? This is what we want to make. Okay? All right, you guys are doing great, I'm sure. Let's get a little reading in, right? Because the test at the end of the year is going to be heavily a lot of reading comprehension. So we want to make sure that we're always making time for literacy and things of that nature. So read and annotate the important parts of the text. So let's read together. What is yeast? Have you ever baked fresh bread? It is a simple process in which a baker combines yeast, sugar, water, and flour together to make dough. However, baking bread does take time. After the ingredients have been combined, the dough needs time to rise. This is where the yeast does its job. Yeast, just like we explored yesterday, is a single-celled fungus. Yes, it is a fungus, guys. Yeast consume the sugar in the dough mixture as their food, producing carbon dioxide as a waste product. The carbon dioxide, a gas, makes the bread rise. When the bread is baked, the gas pockets in the bread give it a fluffy consistency people love. So you can think of that fluffy consistency that people love as being equivalent to that foam that we saw. Both of those are carbon dioxide gas pockets. Okay, now let's head to the next paragraph. What is cellular respiration? So as we stated before, yeast are single-celled organisms. Although humans are multicellular, we undergo many of the same cellular processes as yeast, including cellular respiration. Through cellular respiration, cells generate adenosine triphosphate, also known as ATP. You don't have to know that it's called adenosine triphosphate, but you do need to know the abbreviation of ATP, which is a molecule that stores chemical energy. When the yeast consumes the glucose in the dough, they convert the energy in the sugar into ATP, which they use to fuel their own life processes. This is known as metabolism. In the same way, human cells take in glucose and through respiration, produce ATP molecules to store energy. That's why yeast was such a great model organism for us to use during that investigation because we have so many characteristics in common as it pertains to cellular respiration. They love sugar, we love sugar. They release CO2, we release CO2. They take in oxygen during respiration, we take in oxygen during respiration. So that pretty much checks out. Now, cellular respiration in eukaryotic organisms, these are organisms that have organelles such as yeast and humans, occurs mainly in the mitochondria, right? So cellular respiration takes place in the mitochondria. In prokaryotic cells, such as bacteria, it occurs in the cytoplasm. All organisms use respiration to produce ATP. So all organisms utilize this process to make ATP energy. This is the only way that they're making ATP energy. Anything from plants to bacteria to animals to humans, we all share that in common. Cells break down ATP to release energy in order to carry out life processes, including growth, movement, making molecules, and transporting molecules, right? So we need energy in order for those things to occur. Now, aerobic and anaerobic respiration, these are two different types of respiration, both types of cellular respiration, but there's one primary difference between the both of them. The yeast starts the process of respiration by using available oxygen. This is called aerobic cellular respiration. If you look at the word aerobic, right, you can think of it as aerobic. And if you think of air, you think of oxygen, right? Humans need air because we need the oxygen that air has in it. Same thing with the yeast. So anytime that oxygen is being used, this is going to be aerobic cellular respiration. They have an abundance of oxygen, okay? Oxygen and glucose are converted into ATP, releasing waste products of carbon dioxide and water, right? Just like we had in our input model here, right? We used sugar, glucose, and oxygen, and what we released as a result are the waste products of carbon dioxide and water. So that's just summing up what we literally just spoke about not too long ago. Our own cells use this process to extract energy from the food we eat. That is why we exhale, right? So our own cells use this process to extract energy from the food we eat. And that is why we exhale carbon dioxide and water as waste products, just like the yeast. So we keep coming back to comparing ourselves to the yeast and comparing the yeast to ourselves because we have so many similarities. Next time you hold up a mirror and breathe, you can see the water vapor collecting on the mirror. This is one of the waste products from generating ATP from glucose, right? So if you've ever exhaled on a mirror, you can see that there's some water vapor that is transferred from inside of our body out to the mirror, but it's not necessarily coming out as a liquid, right? We don't exhale liquid water, we exhale water vapor, which is gaseous water, okay? So that's where that moisture is coming from. Now, when the yeast run out of oxygen, they still need to generate ATP, so they switch to anaerobic respiration, which does not require oxygen. So anaerobic means no O2 is required, okay? But they can still make energy. Let's explore. The cells convert the glucose into ATP, generating carbon dioxide and lactic acid as a waste product. However, this type of respiration is not as efficient as aerobic and produces less ATP. Okay? Anaerobic respiration happens in human cells as well. Lactic acid builds up, causes a burning sensation in muscles that often comes with hard exercise. So one distinction that I do want to make here, and this may be a boo-boo on my part, right, is that when yeast undergo anaerobic respiration, they don't produce lactic acid for the most part. They actually produce something else, which is known as ethanol. And ethanol is essentially a product of fermentation, which is a type of anaerobic respiration. And ethanol is an alcohol, right? That's known as drinking alcohol. So they actually use yeast to make drinking alcohol, beers, wines, liquors, those type of things. So just fun fact. But we don't make alcohol when our cells are undergoing anaerobic respiration. We produce another waste product known as lactic acid. That's what's giving us that burning sensation in our muscles, that soreness, that like whenever we're doing push-ups and we get to push-up number 20 and our arms start to kind of feel like spaghetti because it's burning, it's building muscle, but that burning sensation is that lactic acid. And all that lactic acid is doing there is telling our muscles, we need to stop working because we need more oxygen. We need you to breathe in more oxygen to alleviate those symptoms, right? So when we don't have oxygen, it's not the best. We can still do some minimal movements, but it's not preferred that our cells undergo anaerobic respiration all the time, much like the yeast, because as it says here, it's not as sufficient as aerobic respiration and it produces far less ATP from the sugar that we consume, okay? So let's take a look at our Part C exit ticket. What process may cause lactic acid to form? We literally just spoke about this. We can cross out letters C and D. Photosynthesis occurs in plants. That has nothing to do with what we're talking about. And photolysis has literally never been mentioned and it'll never be mentioned again. The only thing that we've discussed is aerobic and anaerobic. And we know that with aerobic, we can think aerobic, lots of oxygen. Anaerobic means a lack of oxygen. And so if lactic acid is being formed, we know that it has to be anaerobic respiration, right? I like to think of lactic acid has a sound bite in it known as lacking, right? Lactic acid, lacking. I would think lactic acid equals lacking oxygen, right? That's how I kind of remember that. And then we look at number 2, it says an individual running a marathon may experience periods of oxygen deprivation that can lead to, right? So deprivation just means a lack of, right? So if we're talking about a lack of oxygen, I know that whatever my answer is has to be anaerobic respiration because we literally just answered it in number 1 and we annotated it in our paragraphs here. So I can cross out anything that deals with aerobic respiration, which is an abundance of oxygen. So that'll be B and D. And now I've increased my chances of 50-50 of getting this question correct. So if I look at letter A, it says anaerobic respiration in muscle cells forming lactic acid. That seems like a very strong answer to me already. But just to check our answer, let's look at letter C. Anaerobic respiration in liver cells producing glucose. Well, anaerobic respiration really might not occur so much in liver cells. It might occur a lot more in muscle cells because muscle cells are the ones that are doing so much work as it pertains to movement and using up a lot of that glucose storage that we have. And we don't produce glucose. We have to eat glucose. It would be pretty cool if we were able to make our own sugar, but our cells can't make our own sugar. If we did, we wouldn't need to eat. We just need to make our own sugar. So I can cross out letter C and A would be the strongest answer here. Okay? And then I am going to just give us like some small notes before we head out, just to kind of wrap this up, right? So aerobic, cellular respiration, I'm just gonna put CR, right? We have oxygen. So O2 plus glucose, right? Both of those things come together. And what it's going to release as a result of that, as waste products, we have CO2 and water as waste products. But as not a waste product, we have ATP energy. And remember, since there's an abundance of oxygen, that means that we're going to make a lot of ATP from that sugar. So we're going to be really hyper because we have lots of oxygen and we have lots of sugar. So we can make lots of energy, right? So I'm going to put a nice, happy, smiley face next to this because my cells absolutely love this. This is ideal, okay? Now, when we're talking about anaerobic cellular respiration, right? We have glucose for sure, but we don't have oxygen, right? Because anaerobic means a lack of oxygen, right? So all we're really kind of using is glucose, and we don't have that abundance of oxygen like we had with aerobic respiration. So I'm still going to produce CO2. I'm still going to produce some water as waste products, right? But now I'm also going to produce another waste product, which we spoke about before, which is lactic acid. That's also now a new waste product, but I'm also going to produce ATP. But the amount of ATP that I'm going to produce as a result of anaerobic respiration is very little. Don't know why that just happened. Very little, right? So if I'm drawing a face next to this one, you bet it is going to be a really sad face, a tear mark, right? This is not ideal for ourselves. We want aerobic to always occur, but we can still do minimal amounts of work on anaerobic respiration, but we can't really do a lot of work, right? So when we think about running a marathon, is that aerobic or anaerobic? You should really be thinking to yourself, okay, well, if I'm going to be running 26 miles, is that going to require a lot of oxygen or is that going to require a little bit of oxygen? And it's definitely going to require a lot of oxygen, right, in order to do that. And then if we're thinking about anaerobic respiration and running a marathon, our muscles will cramp up so terribly without that oxygen that's there. So whenever you think anaerobic exercises, think things that have to be done in spurts, right? In reps. So think of push-ups, maybe crunches, sit-ups, weightlifting, deadlifts, right? Even burpees. You want to think of like things that are going to be like things that you have to like do it and then take a small little rest and then do it again and take a small little rest versus aerobic exercises like swimming, biking, hiking, running. All of those things involve heavy amounts of oxygen and focuses on breathing so that way you can continuously do it without necessarily stopping. Okay, so that's what you kind of want to think about there. Guys, you guys are absolutely wonderful. Our next video is going to literally just do another explore, right? Just like we kind of did with the yeast investigation, except it's a paper investigation. So it'll just be analyzing charts and that will also serve as an explain. So we're going to do an explore and explain right before we get to our elaborate. All right, you guys are super awesome and I'll see you next time. Bye.